Welcome to my class. Today we are going to talk about integrated disease management, which is commonly known as IDM, a very important topic under plant pathology. Alright, I'm going to break down this complex illustration into a part by part format so that it will be easy for you guys to understand the message I want to talk about. Now, before we go into the lesson, let me remind you of what are the main topics that I'm going to talk about. The first one is, why do we need IDM? The second, what is the core essence of IDM? Third, different methods used in IDM. Fourth, disease management components, whether it's tactile, strategic, or a control method. And finally, what are the types of control methods used to control diseases in IDM? Alright, let's talk about why we need integrated disease management. In today's situation, if we are going to only talk about normal practices which are usually used in disease management as a separate topic, trust me, that's not enough. Because if a sustainable agricultural system is to be produced that can be maintained at a particular production rate, we have to come up with a system that identifies the problems involved in each and every such agricultural practice used. For an example, if we are to control a group of fungal infection by only spraying the relevant fungicides whenever we need it, or if we are failing to consider other methods in destroying the particular fungi, trust me, it will be not sufficient. Okay, some factors which creates for such sustainable necessities are mentioned here. Okay, let's see each of them one by one. First one is the resistance of the pathogens. Second one, the emergence of pest resurgence, which is the pest coming back in increasing numbers. A well-known example for this can be given as the resurgence of brown plant hopper in the rice cultivation. Third one, residues of plant materials. And fourth one, the environmental pollution. So a sustainable solution for all these problems can be given by integrated disease management, and that's what we are going to discuss about. So I have used a symbol like this to indicate that it is the symbol of integration, which is to show that integrated disease management is what I'm representing here. And let's see what are the core essence of integrated disease management, because the core essence of integrated disease management is to combine all these kinds of management practices. I mean, I have mentioned many of them, but not all of them, many of them, right? And how to add, integration means basically it means that we are going to add multiple methods in the management of diseases. So that's where the successfulness of the integrated disease management lies, which is to combine all these methods so that an effective input can be obtained. Here I am indicating that all of these methods are used and the arrows can be seen to be pointing out in this direction, all of them, which is to eradicate these kinds of elements, which is either it can be a pathogen or a weed or it can be a pest. So a combination of methods that can be used to eradicate weeds, pathogens, and pests. So that is the core essence of integrated disease management. How are we going to use all these types of elements? With that being said, let me introduce the 5 methods that I'm going to talk about here to manage the diseases. Now we saw why we use integrated disease management and we saw the core essence. And now let's see what are the 5 types of methods that we are going to see today. And the first one is fungicides, the cultural methods, the host resistance, which is a factor which can be used against these kinds of plant pathogens, which is in turn can be divided into 2 other methods as horizontal resistance and vertical resistance. And the host resistance is the factor that we are seeing here. And the next one is the thermotherapy. Thermotherapy is a method where we use heat to eradicate pathogens. The next one is chemotherapy. Chemotherapy is a method in which we use chemicals to eradicate the pathogens. And then organic compounds are combined with mercury to produce effective compounds so that the mercury component of the compound can eradicate diseases. So let's first talk about the fungicides that can be used. Fungicides can be found as either dust or sprays or fumigants. Fumigants are chemicals that can be used to spray and they are injected inside the soil so that the soil can absorb them and produce the effect of a fungicide. So what are the factors we have to consider when using fungicide? The fungicide that we are going to use should be less toxic so that they can be less harmful to the people who are going to eat these plants, so that can be useful. And the fungicide that we are going to use should be crop specific, because we should only target a specific crop. So the fungicide that we are going to use should be crop specific, or else it's going to be a problem for human consumption, because if we are going to use excess fungicides that are not crop specific, there will be other issues. Then the dosage, the amount that we are going to spray, because if we are going to spray fungicides in unnecessary amounts, it's going to cause an economic problem. Then the timing, at what time we should use these fungicides to crops, because if you are going to use this like in unnecessary times, then that's going to be a problem as well. And how many times you are going to apply these fungicides, and then it should be not carcinogenic and it should not be mutagenic, which means it shouldn't cause cancer, and mutagenic, which means it shouldn't cause any kind of gene mutations, so there will be alteration in their genes. And finally, the interval between the last application and harvest should be considered. So all these factors should be used when considering the use of fungicides. Only then an effective use of this fungicide can be ensured. Okay, the second factor we are going to see is about the cultural methods that we are going to use. So I have used the culture plate as a symbol to indicate what's going on here. There are about like 4 factors that I want to consider here. So we are going to consider about the irrigation practice that we are going to use. The irrigation practice can be either drip or versus overhead. Overhead practice is a method which is used usually to sprinkle water in a hole to the plant. Drip is a method in which the water reaches the plant specifically. So it doesn't reach as a spray. It goes to the plant as a droplet, so that the plant will get the enough amount of water, only the amount of water it needs, and not any more. The second is the timing of planting also should be considered, because at what season you are going to plant and at what time you are going to plant. And there should be wider spaces between the rows. And if we have high moisture content always favors high amount of pathogens. And that's something which is already known. The first 3 that I have mentioned in these cultural methods are really about moisture management. And moisture management is very, very important in integrated disease management. So the third factor that we are going to see is about host resistance. Host resistance is an important factor in disease management. There are 2 types of host resistant practices. We call that as either horizontal resistance or vertical resistance. So these are 2 types of host resistance that can be seen in nature. So the first one is horizontal resistance. In some textbooks we can see this being called as either field resistance or partial resistance. And second factor is vertical resistance. Now let's see what is horizontal resistance. They are polygenic. Now what does this mean? Horizontal resistance, it's a kind of resistance where a certain group of plants show resistance against all kinds of pathogens. Second one, vertical resistance. So vertical resistance can also be called as major gene resistance or R1 gene resistance according to some textbook. And the difference between vertical resistance and horizontal resistance is that vertical resistance is monogenic, which means it's only show resistance towards one kind of pathogen. So here is a bar chart showing 4 different types of resistances. So here is an example of 4 different types of plants in which the resistance is shown in y-axis. The x-axis shows the type of pathogen race involved. It is like pathogen race 1, pathogen race 2, and so on. Now let's analyze the 4 types of graphs here. These graphs denote the amount of resistance shown by each pathogen race in a bar form. The first one, ANC, which shows no vertical resistance. To understand what does this mean, let's see the second one too. BND, which shows vertical resistance. Now, how does ANC differs from BND? ANC shows pathogen races which have equal levels of resistances. But if you see BND, the pathogen races are different. The resistance shown by each pathogen differs within themselves. If there is a difference in the resistance shown by each pathogen, then those graphs can be said as they are showing vertical resistance. So remember, if the resistance shown by plant is the same against all type of pathogen races, then the resistance is said to be horizontal resistance, which is polygenic. Let's talk about 2 graphs A and B, how they differ from C and D. Now that's another perspective of seeing how these graphs work. Okay, if you look at what I have written here, it says A and B shows moderate level of horizontal resistance, while C and D shows low rate of horizontal resistance. A and B shows moderate level of horizontal resistance and C and D shows low level of horizontal resistance. Now, how do we understand that? We understand that by seeing the point in the graph that shows the lowest level of resistance. Now, if you look at A and B, you can see that this level is same as this level. The lowest point in this one shows the same level of resistance as this point. But if you look at C and D, the lowest level is below that, which is the lowest point in C is same as that of the lowest point in D. So that's how we find which shows the lowest rate and which is moderate rate. If we go more higher, then it will be called as A and B having a high level of resistance. Now another factor that we have to note is the host genetic diversity is important to slow down epidemic. Now why is that? If we start sending the same type of host for a particular pathogen, then the problem is that the pathogen will overcome the host resistance and it will easily spread. But if we have different types of hosts, then it will be easy for the host to attack the pathogens, because when there is genetic diversity, in other words, the crop plants don't attack the pathogens as same as the tall plants, right? So there is a difference between them. Another point that we have to note is, you may ask that how does horizontal resistance differs from vertical resistance? If a plant can only attack pathogen with only a certain type of gene, there must be a difference between horizontal resistance and vertical resistance. So there is a difference that we have to understand, that vertical resistance reduces the inertial amount of inoculum that's available. So that's an additional thing that is found in plants with vertical resistance. Now let's see another type of factor that helps in integrated disease management, that is thermotherapy. Now thermotherapy means we are providing heat for the eradication of a particular type of pathogen. So these are the factors that we have to consider about thermotherapy. In a certain type of thermotherapy, we provide heat for the soil so that the soil can be sterilized from the pathogens. Now this heat is provided by pumping steam. So that's a method that can be used. And then you can use hot water treatment for the planting material, because if we have to sterilize planting materials before we are going to plant some kind of crop, because you can get any type of pathogen from any area. Pathogen from any area can infect the plant that we are going to plant. So we have to sterilize these kind of materials before we are going for a planting program. And next, another type of thermotherapy is solarization. So we leave the plants in an open field so that we can eradicate the pathogens. Or refrigeration. So you know that the metabolism of organisms are lower at lower temperature. When we refrigerate harvesting material for a certain amount of time, what happens is the metabolism of the pathogens slow down. So refrigeration is another option for treating planting materials. So by using refrigeration, we can't completely eradicate planting material from spreading diseases, but we can only slow down the diseases that are spreading. So that is also an effective method in controlling diseases. So all these factors should be considered when you are going to plant a seed or a stem or a root cutting. So we have to consider all these factors, and that's why we consider this type of management as integrated disease management. We are going to consider all these factors when we are going to plant a material. So this is chemotherapy. Now I have shown symbol to denote that it's a chemotherapy. That's a symbol of a saline bottle. Actually, in chemotherapy, this name itself suggests that we are using chemicals to treat the planting materials. We are using chemicals so that we can eradicate a type of pathogens or weeds even. Now let's see some of the factors involved. Now let's see some of the points that we can see under chemotherapy. Bactericides or anything that ends with the suffix "cide" can be used in chemotherapy, because that's obvious, right? I haven't mentioned fungicides here because we have discussed about it earlier itself as a separate topic. That's one thing. And then plant antibiotic. Okay, different types of plant antibiotics can be used. Alkylating agents, or for an example, it's metal salts like heavy metals like magnesium chloride, aluminium chloride, and so on. Then there is another factor, that is antimetabolites. Antimetabolites are substances that can be used to slow down chemical process, the metabolic activities which happens inside cells. So that makes sense, right? These are the factors that we can consider as components involved in chemotherapy. So these are also some factors that we have to plan before we are going to apply them to the plants. And finally, let's talk about organomercurial amendments that can be used to control the diseases or the plant pathogens involved. So I could have considered it under chemotherapy, but to make it prominent, I have showed it as a separate topic. Mercury can cause serious damage in plants, so it is used as fungicides for seeds and plants. An example for this, a record which includes ingestion of flour made from ethyl mercury treated seed grain. It has happened before, so that's an example for it.